Right, here we go. Mock exam talk through question 2. We'll be scrolling up and down for this one, just like you had to when you did your question 2. The first one is you are given an identification key for your different bacteria. And the first things to consider is, what do you actually know? What's the difference between a coccus and a bacillus? That is something you should know, that a coccus is more spherical in shape and a bacillus is a rod-shaped bacteria. And from there, it's a question of just going through them, and most of you did use that key really well. So if you've got one correct, you will have all 4 correct because you figured out how to use it. So let's go through the first one. The first one was probably hardest. Coccus, because it's a round shape. It doesn't look like it's got any particular lines within it, sort of chains of one cocci attached to the next ones linked together. So they're all more clumped, more lumpy. You know it's gram-positive and it's a facultative anaerobe. So let's look at what this could be. So it's gram-positive. That means it's on the left-hand side. It was a spherical shape, so it's a coccus. It can't be on that side. You've got a facultative anaerobe over here, but it can't be that one. You have got a facultative anaerobe which comes as clumps, not as chains. So that would be a staphylococcus, a general infection, staph infection one. The second one here, B, it's a rod, it's a bacillus. It could potentially be linked together, but let's have a look at what we get to. Gram-positive, facultative anaerobe, and it's a rod shape. So gram-positive, we're on that side, it's a bacillus, and the facultative anaerobe. So here we go, we've got a bit of yogurt, a bit of a lactobacillus making the making. Looking at C, spherical, therefore a coccus. It looks lumped rather than distinct chains. Gram-negative and facultative anaerobe again. So let's have a look. Gram-negative, so on the right-hand side, it was a coccus, and you've only got one facultative anaerobe, which means you've got potential gonorrhea or meningitis in her making here. And the last one is a rod-shaped, possibly linked together as a chain, gram-negative facultative anaerobe. So gram-negative were on the right. It was a rod-shaped, so it's a facultative anaerobe. There's only one that's facultative anaerobe, which is our good old lives in the lower intestinal tract, E. coli. Here we go. As mark scheme, it literally looks like this. And most of you did this one really well. So moving on swiftly. This one, as opposed to the first one, was done really, really badly. If you have a D value, and this question is, we're doing it for you here, do it yourself here on that temperature. We've done a procedure with the 72 degrees temperature. You want to do that procedure at the 62% temperature. Let's have a look. A D value is the time in minutes. So we need to get to some sort of time. That's going to be our final answer, has to be some sort of time. The time in minutes for a given temperature, which is either this temperature or that temperature, to destroy 90% of a population. So if you had 100 bacteria beforehand, you would have 10 afterwards. If you had 1,000 bacteria beforehand, you would have 100 afterwards. If you had 10,000 bacteria beforehand, you would have 1,000 afterwards. Hold on a second, have a look at your logarithmic scale. That literally means 10 to the power of 1, 10 to the power of 2, 10 to the power of 3. So that would be 1,000. That would be 100. So if we're making that one step from 1,000 to get to 100, we have lost 90% of the population. In this case, they've decided to do it from the 10,000 to the 1,000. So here from 4 to 3, you've lost 10,000. Trace your lines across, trace your lines down. What is your time difference? Okay, so it has been done for you here. The D value here is 1 minute, so from 2.3 till 3.3. Now, let's have a look at, you are meant to do this here. You can do it for any consecutive value. If you're going from 5 to 4 or from 2 to 1, you'll still get the same D value. You'll still get the same time interval. On the mark scheme, they've chosen to do it for 4 and 3. If you go from 4 across and then down, you get to, I think it was 23. From 3 across, you get to 36. Let me just look at that mark scheme. Yeah, 36, take away 23. And then you get to your D value of 13 when you're counting them out. The actual doing it is easy. The understanding the question is hard. Okay, so that's why the talk through. Then this question here, in a nutshell, says, can you describe how to do serial dilutions? 4 marks, let's go. So serial dilutions, in a nutshell, what you need to have is that you're starting with a sample. You're gradually reducing the concentration, always 1 mils to 9 mils, 1 mil to 9 mil, to lower the concentration in a specific way. Then the plating, you never plate more than 1 mil so that it doesn't drip off your agar jelly. It does say like no reference to aseptic technique, but the spreading is something that could be there. Counting colonies. Oh, actually, first we need to incubate that baby. So you're doing your serial dilutions. Describe how to set up your different serial dilutions. Plate 1 milliliter. Then I have had various lovely ideas on how to incubate it. The standard is between 24 and 48 hours. And in a hospital lab, you'd kind of grow them at around 36, 37 body temperature. However, in a science lab where you don't want to grow anything too harmful, you would do it at a much cooler temperature. And what is suggested here is that you kind of use a lower possibly as well. Now, it would be acceptable to go all the way up to 37, but in a school lab or college lab, we would stick to around 25 rather than the more dangerous temperatures, which would grow body temperature pathogens in there as well. Okay, counting. Some of you said count it. I can't count an individual bacteria. You need to have the word colonies there. You need to have the colonies there. Once you've got your counted colonies, it's the mathematical way backwards. So you are using your dilution factor as well, again, and multiplying by that the opposite of the dilution factor to get to your actual number of live viable count, the live bacteria that you've had. Repeating the whole procedure and counting the means, calculating the means would have given an extra mark here. So any 4 from those I would have had to have in there. You should have the mark scheme available, otherwise you can just pause that on this slide. Okay, then the effect of heat in the process of pasteurization on the proteins. The key words here are heat and proteins. This is not a question of bacteria, this is biochemistry. This is a question as to what type of bonds are being destroyed when you've got protein structures. The key ones that are destroyed are the hydrogen bonds. The hydrogen bonds will not make it past different temperatures. Also pHs destroy hydrogen bonds, but temperature is a key one. Therefore, you're disrupting the 3D shape, both the secondary and the tertiary structure, as well as actually the quaternary structure as well. You're disrupting the entire shape. Once the shape is no longer there, proteins no longer fulfill their functions, enzymes no longer fulfill their functions, and bacteria may not be able to complete any of their metabolic pathway and they'll die. So if you look at the mark scheme, it does speak, you need to be clear on heat or kinetic energy breaking those bonds. Kinetic energy breaking the hydrogen bonds. Denaturing by itself, I can't take. It does need to be in a bigger picture here, please. Then what would those hydrogen bonds have done? They would have held together the 3D shape. They would have held an active site in the shape that it needs to be, for example. Therefore, the protein can no longer produce its function, and eventually you're going to have the death of your bacteria. Right, that was question 2.